Hi class and welcome. We are now on the third topic for the subject Bio 108. In this lecture video, we will discuss sensory and muscle physiology. So what is a sensory receptor? A sensory receptor is a structure that reacts to a physical stimulus in the environment, whether it's from the internal or external environment. Here, the sensory nerve ending receives information and conducts a process of generating nerve impulses to be transmitted to the central nervous system, particularly the brain, for interpretation and perception. These receptors are usually found in the sensory organs such as the olfactory receptors in the nose, taste receptors on the tongue, and light receptors in the retina of the eye. There are 6 basic types of sensory receptor which are classified based on the type of stimulus they perceived. These are chemoreceptors, thermoreceptors, mechanoreceptors, photoreceptors, magnetoreceptors, and electroreceptors. These receptors can also be classified whether they are found internally or externally. First is enteroreceptors, which monitors ionic composition of the body fluids internally, and exoreceptors, which monitor external conditions. So the main function of sensory receptors is to act as transducers. That is, they convert or transduce energy from one form into action potentials. So for example, stimuli perceived from the temperature and light energy are converted into electrical activity. For the animal to perceive any sensation, the local potential must be converted into action potentials, which are considered to be the currency of the nervous system. In the case of free nerve endings such as Pacinian corpuscle, which is an example of a vertebrate pressure detector, the local potential, which in this situation is called a receptor potential, is transmitted by local currents to the first node of Ranvier. If the change in membrane potential is sufficient to bring the membrane to a threshold at the first node, an action potential is generated. In the case of sensory receptors, which are specialized cells, the local potential, which in this situation is called a generator potential, results in the release of neurotransmitter. The neurotransmitter is then able to influence the electrical activity of the neuron associated with the sensory receptor. In this case, when the neurotransmitter released in the synaptic cleft will bind to the receptor, the channel will open, allowing ions, for example, sodium, to enter the cytosol and will influence the activity of the neuron, causing an action potential. When subjected to a maintained stimulus, sensory receptors respond by decreasing the frequency of action potential production. This phenomenon is called adaptation. It is a form of integration. So some receptors may adapt very slowly, and this is called tonic adaptation, while others may adapt very quickly, or what is known as phasic adaptation. Now, reception is a process of activating a sensory receptor by a stimuli, while the process, again, is known as sensory transduction. Chemoreception is the detection of specific chemicals by a sensory receptor. This includes the sense of smell and taste, which play important roles in nature. Another type of sensory reception is mechanoreception. It is the measurement of force or pressure and displacement. The mechanism involved is that stretch-activated ion channels, which are found in the membrane of the receptor, are closed at rest. However, when stimulus or pressure is applied, a conformational change in the protein of the ion channel will occur. This leads to depolarization and the generation of local currents. So now, effectors are the responses to sensory information. Effectors can be defined as anything capable of producing a biological response. This can range from a muscle producing movement to endocrine gland secreting hormones to alter aspect of metabolism. The most obvious response that animals make is that of movement, which is governed by muscles. Muscles are made up of many muscle fibers equivalent to muscle cells. The cell membrane which surrounds the muscle cell is called the sarcolemma. It has a number of invaginations into the cell called the transverse or T-tubule. The cytoplasm of the muscle cell is called the sarcoplasm, while the endoplasmic reticulum of muscle cells are called sarcoplasmic reticulum. Muscle fibers in turn are composed of myofibrils. These myofibrils are seen to be banded in appearance. Some of the bands, such as I band, appear to be lighter than other bands or A bands. This is because I band contain only the thin filaments or actin, whereas A bands are regions where thin and thick filaments overlap. This thick filament is known as myosin. Each I band is split into two by the presence of a Z-line. From one Z-line to the next Z-line is known as a sarcomere, which is the basic unit of muscle contraction. So in turn, the myofibrils and therefore the sarcomere are composed of two myofilaments. The thick filaments, which are composed of protein myosin, consist of two polypeptide chains with their myosin head, while the thick filaments are formed from two actin molecules coiled together with the two tropomyosin molecules wrapped around them. Pairs of troponin molecule occurs at interval along the actin molecule. So how then does contraction occur? The mechanism by which muscle contracts is known as the sliding filament model. With this model, the actin and myosin filaments slide past each other without physically shortening. So at rest, when the muscle is relaxed, the protein, troponin, and tropomyosin prevents the formation of the cross bridges that are necessary for contraction. During contraction, the filaments slide past each other, resulting the Z-lines to move closer and the muscle shortens. The contraction of skeletal muscle is initiated by the release of a neurotransmitter. In the case of vertebrates, this is the release of acetylcholine from the axon terminals of motor neurons. The effect of depolarization is to release calcium from the sarcoplasmic reticulum where it is normally stored at rest. Calcium released into the sarcoplasm binds to troponin, inducing a conformational change. This will cause a change in the orientation of the tropomyosin molecule, exposing a binding site for myosin in the actin filament. The exposure of the myosin binding site allows the two myofilaments to bind to each other, forming the cross bridge. Remember that the cross bridge formation is the initial stage of contraction. The head of the myosin converts ATP to ADP, an inorganic phosphate. In doing this, the energy is released, which is stored in the myosin head. Its effect is to pull the thin filaments towards each other, shortening the length of the sarcomere and causing the muscle to contract. During the cross-bridge formation, the ADP and inorganic phosphate are bound to the myosin head. The removal allows the bonding of another molecule of ATP. This causes the cross-bridge to cease. The molecule of ATP is broken down to ADP and inorganic phosphate, leading to the reformation of cross bridges. The cycle will continue and the microfilaments are pulled past each other, causing the muscle to contract. So the whole process is terminated when repolarization of the muscle cell occurs. During repolarization, calcium is removed from the muscle cell and is stored in the sarcoplasmic reticulum. This allows the tropomyosin molecule to return to its original position on the actin molecule, thereby blocking the myosin binding site. So that ends our discussion for the topic on sensory and muscle physiology. If you have questions and in need of clarification, please feel free to contact me or post it in the Questions and Concerns forum in our Mole classroom. Thank you and see you again in our next session.